Good morning. In this presentation, we would like to talk about the implementation of poliovirus environmental surveillance in Hungary, which takes place in the National Center for Public Health and Pharmacy. Polioviruses are single-stranded RNA viruses in the genus enterovirus. It's transmitted by person to person, mainly through the fecal-oral route. The majority of the infections are asymptomatic. Acute flaccid paralysis, or AFP, occurs in less than 1% of all infections, and it mainly affects children under 5 years of age. In 1988, the WHO launched the Global Polio Eradication Initiative. The GPIE uses two types of vaccines, an inactivated one and a live attenuated oral polio vaccine. With national vaccination campaigns, the incidence of polio has decreased by 99%. There are only two endemic countries left, Afghanistan and Pakistan. The failure to stop polio in these areas could result in the global resurgence of this virus. Since 2002, after declared as polio-free, in the European region, several cases of wild and vaccine-derived poliovirus cases has been monitored. That is why AFP surveillance as the gold standard in ad hoc environmental surveillance in regions declared as polio-free are important. During the COVID-19 pandemic, around 23 million children has failed to receive basic childhood vaccination. As polio vaccination is among the basic vaccination, basic immunization procedure, this statistic also reflects the lack of immunization against polio. With this event, it has accompanied with an increment of emergence of either wild and vaccine-derived poliovirus cases. Between September and December 2024, in the European region, 5 countries' surveillance system detected circulating vaccine-derived poliovirus 2 strains. Although no related human cases were found, this event highlights the importance of high vaccination coverage and in environmental surveillance system. So, since January 2024, routine testing for poliovirus in wastewater in Hungary is occurring in accordance with the WHO guidelines. It is The surveillance system is operated by the National Center for Public Health and Pharmacy in cooperation by 2 units. In the next 2 slides, I would like to talk briefly about the tasks of the Environmental Health Unit of the Public Health Laboratories and Methodology. Samples are taken from 9 sampling locations. 3 is in Budapest, 3 wastewater treatment plants in Budapest, from the Ferenc Liszt International Airport, and 6 county seats with more than 100,000 inhabitants. The sampling is carried out by trained wastewater treatment plant staff, and it transported through the National Wastewater Monitoring System for SARS-CoV-2. 2 samples are tested weekly in a rotating system. After centrifugation, a pellet extraction with chloroform is performed, and the water phase and the supernatant from the centrifuged sewage sample is concentrated by ultrafiltration. Around 1 milliliter of this concentrate is used for the pan-entero and pan-poliovirus PCR. And during the validation phase, we found that the poliovirus RNA recovery efficiency, in the RNA recovery efficiency, there is around one-fold difference between the spike-Swiss sample and the viral sample. We also found that the limit of detection for digital droplet PCR is around 2 times 2 in the power of 2 and around 1.7 times 10 on the power of 3 genome copy per liter for qPCR. The other 1 milliliter of this concentrate is provided to the National Biosafety Laboratory, and I would like to ask my colleague Nora Dézsi Magyar to continue this presentation and talk about this in detail.
Thank you, Bogi. So I would like to introduce you to our part of this surveillance project that we are running at the National Center for Public Health and Pharmacy. I'm from the National Biosafety Laboratory, which serves as a polio essential facility in Hungary, and I'm also working at the National WHO Poliovirus Reference Laboratory. So we are mainly doing reference activity in order to prove the polio-free status of Hungary in the frame of the global polio eradication. There are 3 main surveillance programs that we are running. The first one is the AFP, which stands for acute flaccid paralysis. So basically, we are screening children under the age of 15 years old, presenting with paralytic symptoms in order to exclude poliovirus as a cause of infection. We are also running the environmental surveillance program that we are talking about today, and also we are running the enterovirus surveillance program, which is case-based in our laboratory, and we are doing that in the frame of the NPAN network. Our laboratory is accredited by the WHO, and we have to prove our competence annually by different proficiency tests. So if we receive a sample for either AFP or environmental surveillance, we have to test the sample by virus isolation. This is mandatory. And report the results towards WHO in a timely manner. And if there is a suspicion for potential poliovirus introduction into the country, we have to do intratypic differentiation by RT-qPCR, and either we can use Sanger or next-gen sequencing to confirm the results. There are 2 different cell lines prescribed by the WHO, which we have to use for virus isolation. The first one is the human rhabdomyosarcoma, or RD cell line. This is susceptible for basically all enteroviruses, including polio. And according to the WHO standards, a laboratory is running sensitive surveillance if over 50% of the tested sewage samples are RD positive. And the other cell line is a mouse-derived transfected cell line, expressing the human CD155 receptor, which is used by poliovirus during infection, and this cell line is selective only for polio. This is the very nice algorithm that we must follow in the laboratory. This is, as you can see, very demanding, but we have to report each and every step towards WHO. What I want to highlight here is that if we found one sample to be L20B positive, we must handle that sample in high containment according to the GAP4, the Global Action Plan for Poliovirus Containment rules. So we are working with that in a biosafety level for a containment. I actually stole this slide from the WHO. This is the result of the 2023 performance in the European region of the WHO PolioRef labs. What interests us here is the lower figure, the showing the environmental samples. As you can see, around 20% of the samples were L20B positive in 2023. But I can tell you that none of these samples were either a white-type polio or vaccine-derived back-mutated poliovirus. It was Sabin or a polio strains from the vaccination. And as you can see, the European region reached only 33% of non-polio isolation rate, which is a little bit below the standards. This is our results from the first year of implementation. During the first year, we succeeded in 53.3% of the cases. In terms of non-polio isolation, it was significantly higher in the summer and autumn months, but it was not surprising when we compare the data with the number of confirmed human non-polio cases. Basically, the two data correlated very nicely to each other. And in April, we found one L20B positive sample in Budapest, so we followed the WHO guidelines on classification and reporting towards WHO, and we performed the intratypic differentiation qPCR and also whole genome sequencing from the cell culture supernatant, and it turned out to be a Sabin-like 3 strain, so it was basically just one mutation distance from the oral polio vaccine, type 3. And just very briefly, as kind of like a take-home message, I'd like to highlight the importance of the poliovirus environmental surveillance and kind of link it with biosafety. So this is serving as an early detection tool for poliovirus detection, and sometimes even before the clinical cases are identified, which makes us enough time for proper intervention and countermeasures, for example, vaccination campaigns. And not just at the national or wide-scale level, but also at the laboratory level by testing the sewage, we can track inadvertent release of poliovirus. And I think it's a very good example of a nice cross-disciplinary or cross-sectorial collaboration between scientists and professionals from the different fields. Yeah, and with that in mind, thank you for your attention.